Hi, I'm Lindsay Williams. I'm also from Michigan State University. And I've also been working at the same system that Mary was just speaking about today, Chromo. And I'm going to be talking to you today about some very preliminary work that is a bit of a stepping stone in order to look at seasonal and episodic microbial community dynamics within Chromo. So as Mary and a few others have pointed out throughout the day, serpentinization is a very important process for creating chemosynthetic processes that microbes use to live. And she also introduced our field site, so I'm not going to touch on that very much right now. But in terms of my analyses and what I'll be talking about today, I am focusing on the QV11 and the CSW11 wells that are located at these two different well clusters that Mary mentioned, and they are both the uncased wells. So the Chromo wells, 8 wells were drilled in 2011 with the purpose of monitoring the geochemistry, the microbiology, and the hydrogeology of the site. And thus far, some of the prior research has indicated that there's low microbial diversity and has also indicated certain substrates that can be used to successfully culture some of these organisms. And it has also indicated organisms that primarily occupy the deep anoxic source waters, as well as oxic and anoxic interfaces. So the thing that I want to really focus on in terms of this site is looking at seasonal dynamics. So we've all experienced seasons here on Earth, but we do also know that other planets and moons can be subject to seasonality if they are properly oriented. And so if they are subject to seasons, this is going to change conditions that any life that may live there is going to be experiencing. And so we have indicated either evidence or potential evidence of serpentinization on different planets, moons. And so we've also noticed within our own serpentinization sites at Chromo that certain microorganisms have numerous genes that allow them to basically use a variety of different substrates. And this can indicate that they might need to be able to use those different substrates because something about the environment is changing and basically requiring that they use different things in order to live. So the things I wanted to address here were what factors affect microbial abundance overall based on all the samples that we've taken throughout the time there at the site. Are patterns of cycling seen in microorganism cell abundance and other geochemical parameters? And do different factors play a larger role in microbial abundance in different environmental conditions? And so to begin to look at this, there we go. I wanted to analyze some data that we had on the water table. So this is data that we've collected starting in 2014, and it spans through to the summer of 2016. We had in situ pressure and temperature transducers installed in these walls, and they basically take hourly samples of water table elevation and temperature. And so other than times that we were there and pumping, which you can see the drawdown in the water table, there does seem to be some evidence of cycling, probably due to drought, increase or decrease rain in the area. These are two different plots. The one on the left here, this x-axis is time, and then up here we can see cell abundance in 10 to the 5 cells per mil, and below here is the depth of the water table in meters. And so at CSW11, the water table seems to be relatively stable, but we do see some visual evidence right off the bat that there does appear to be some cycling in the cell abundance. And over here, again, I have the x-axis as time, and I wanted to put all the geochemical and other parameters that we measure for on the same graph. So some of the units are weird, but you can see what units each of these are in. But essentially what we see is that we're seeing some cyclic nature with ORP, dissolved inorganic carbon, dissolved methane, and sulfate. These are the same exact plots with the same axes, and generally the same parameters here for QV11. And we can see here that there's a little bit more variability within the water table. We're still seeing some of that cyclic nature in our cell abundance, and we're also seeing a cyclic nature in ORP, dissolved methane, and dissolved inorganic carbon. So these are all promising in terms of looking at seasonal effects. So what I wanted to do with some of this data was try to figure out what things are most important in predicting what the cell abundance is. So to quickly look at this, what I wanted to do was create multiple linear regression models using the data that we have. We do have some gaps that were in our data set as we weren't always able to sample for everything. So what I did to start was compile all my data and essentially simulate my missing time points using Markov chain Monte Carlo permutations, which is a method that has been used successfully in other biological applications. The next thing that I did was set up multiple linear regressions, setting my cell abundance as my Y value, and then essentially comparing it to a variety of different X values that would be my different geochemical parameters. And so essentially what I would end up with is my cell abundance with an intercept, and then plus each of those different geochemical parameters. And initially I started out with every single variable and then manually removed them in a stepwise manner to obtain a model that had significant coefficients overall. So I started with a full model to use all the time points that we have, and for CSW11 and QV11, these are the parameters that were deemed significant in the model, and you can see the adjusted R² down here. And the check marks that are displayed next to some of these parameters are they're ones that were indicated as or had a clearly visual cyclical nature in some of those plots that I showed you earlier. So following this, what I really wanted to look at is seasonality. So I needed to determine how I was going to separate my seasons. We do know that the Lower Lake County, which is where our site is located, is subject to flooding in the winter and then some drought in the summer. So it appeared from these graphs and this data that from November through to March, there seems to be significantly more precipitation in the system, and that from April to October, it's in a drought stage. So I chose a wet and a dry season, respectively. I followed the exact same model, basically, to create these wet and dry models in doing multiple linear regression. And so these were my parameters that came up as being significant for CSW11 for the wet and the dry season. And Valerate, which has a star next to it, was a parameter that was not initially in that full model when I used all the time points. So that indicates that Valerate may be important in the wet season. I did the same thing with the QV11 wells, and again here you can see that the depth to the water table and then specific conductance in both the wet and dry season seem to be parameters that are maybe more important within these different seasons as compared to overall with my full model. So some conclusions from this initial analysis is that it does appear that unique parameters that are different from what the overall model that seems to predict cell abundance were found to significantly contribute to cell abundance. So that does seem to indicate that seasonal effects on the system could be playing a significant role in microbial cell abundance. And so in order to further investigate this, I'm going to be incorporating 16S rRNA data in order to provide more robust results as well as indicate effects on dominant species in the systems, and then do further regression and clustering and other analyses in order to better visualize any trends that we see here. In terms of implications, understanding how microbial communities respond to changing environmental conditions is important in order to understand what taxa might be more prominent in a specific condition. And so based on this, if we're visiting an astrobiological site that seems to be suitable, this kind of knowledge could be important for indicating geochemical parameters that might be more important to test for, metabolic functions that could be occurring, and also inform on culturing methods that might be more successful. I would like to thank the Strength Lab Group, as well as NAI-RPL for funding this work, and our collaborators for all of their help. Thank you.
All right, we have about 3 minutes for questions, so come up to the mic if you have a question.
Do you get the same in analyzing your seasonal patterns? Have you tried using actual climate data for those time periods as opposed to annual and monthly averages?
So I have looked into that. Part of the issue, well, not issue, but I guess I didn't really get the time to go back through some of the weather data and actually pull temperature, precipitation data from the exact dates that we were there sampling. So that would actually be a really good thing to incorporate as I continue with this.
There's been some pretty variable years in the past 5, so
yes,
you might get some interesting results.
Yeah, which could potentially help by narrowing it down.
Any more questions?
Yeah, this is Dave DeMaria. Maybe you said this, but it seems to me that the depth of the water in the well and also just the chemistry of the surface, the upper part of the well, that's really what would be responding to seasons and maybe what really drives the results that you see. And I presume you did that, but I just don't remember.
In looking at the chemistry?
Well, like during the winter, you'd have more fresh water coming in.
Right.
The well would be higher, and that would affect this gradient going down the well. So I presume you made those connections, but I just...
So all of the samples, essentially, both the microbiology as well as any of the geochemistry samples, for my purposes, were all taken from water pumped from the very bottom of the wells. So even though there is more water coming in the system during the winter, there would probably be a lag, but essentially we'd probably see the influence of that meteoric water coming down through the surface affecting those communities potentially when it's rainier.
Right, thank you.
Thank you.